Today we will discuss another topic, that is bioremediation technology. Basically, there are various aspects in bioremediation, and I'll give you an overview on the different types of bioremediation technologies that is available. So, the term bioremediation, it can be explained in this manner, like it includes two terms, that is bio and remediate. So in biology, remediate is actually the word meaning is to solve a problem. So if you are biologically remediating or solving a problem, then that is bioremediation. Usually, bioremediation involves is the use of biological organisms to solve some environmental problems. So that is the meaning of the term bioremediation. Bioremediation is a collective range of cleanup methods by using natural microorganisms such as bacteria, fungi, etc., to degrade hazardous organic contaminants or convert hazardous inorganic contaminants into environmentally less toxic or non-toxic compounds of safe levels in the soil. Also, not only soil, subsurface materials, water, sludges, and residues. That means that definition includes various items. Like it's a collective range of cleanup measures. It is not a single method, it's a collective range of cleanup methods using natural microorganisms such as bacteria or fungi. And it will degrade hazardous organic compounds and inorganic contaminants such as heavy metals and other things to environmentally less toxic or non-toxic levels. It doesn't assure that it is completely or it will remove all the pollutants from the environment completely. Maybe it will reduce that toxicity to a less toxic level. So that contaminants can be environmentally or it is converted into environmentally less levels or non-toxic compounds of safe levels in the soil, subsurface materials, water, sludges, residues, all other environmental sources. So in all these cases, like soil, subsurface materials, water, sludges, and residues present in the environment, these contaminants will be reduced to less toxic or non-toxic levels by the action of different natural microorganisms like bacteria and fungi. So that is the meaning of the term bioremediation. So the hazardous organic compounds or organic waste that includes petroleum products, fungicides, insecticides and herbicides. All these things were widely used as a part of agricultural practices and other things. Fungicides, insecticides, and herbicides. And there is the petroleum products that is also nowadays commonly used in various forms. So this contributes to hazardous organic waste, complex organic compounds that is present in the environment that is contributing a significant quantity of the pollution. Then the most common contaminants in this category, they include commercial hydrocarbons like gasoline, diesel, jet fuel, naphtha. These are all different forms of the hydrocarbon fuels. Then domestic heating oils, which is used in your day-to-day life, different forms of oil and which is washed out of your sink. Then chemicals like BTEX compounds. BTEX is an abbreviation for the terms that is benzene, toluene, ethylene, and xylene. These are the chemicals which is categorized as BTEX compounds. These also contribute to the environmental contaminants. Then organohalogenated compounds like different solvents, including trichloroethane, tetrachloroethane, etc. Then heavy hydrocarbons like crude oil, pipeline or tanker, rail spill, or oil spills, uh, heavy fuels from electric plants, tar, etc. And caustic from that is used to used in wood treatments. That's different forms of heavy hydrocarbons that is nowadays used for and it is actually contributing environmental contamination. Then different heavy metals, explosives. Okay. All these are different categories of the common contaminants which is present in the nature that require some form of a bioremediation. Then the remediation methods that includes biological methods, which may include biotransformation or biomineralizations. That is one term. And another one is chemical remediation methods, in which these compounds were destroyed or fixed or neutralized using chemical measures. Physical methods like concentration, excavation, or and other methods like that. Then solidification and encapsulation and removal, subsequent removal. Vitrification at high temperature by fusion and other things. Then thermal methods including incineration, gasification, pyrolysis, etc. So there are a lot of methods for remediation of such contaminants and other things. But our focal point as microbiologists will be based on biological methods, which involves transformation, mineralization, and other things. These transformation includes biotransformation or the transformation of a compound or a complex compound into much simpler forms that can be further utilized or degraded by subsequent organisms that is coming in the succession. And mineralization, it is actually a distinct term when we compare it with bioremediation, which is a general one, or biodegradation. Biodegradation means it is a conversion of a one compound A into B by the action of some biological entities. So the compound A is converted to B, which may be a further toxic compound or a more toxic compound. So that doesn't mean that it is completely neutralized. The toxicity is removed and the environmental threat is or hazard is removed by converting from A to B. Because the B may be a more potent or more toxic compound during the degradation process that is produced. So the biotransformation or biodegradation, different forms of biodegradation doesn't ensure that the toxic compound is successfully removed. But when we consider the term biomineralization, mineralization is actually, it reaches the end product, that is for example water or oxygen or something, CO2, something like that. So if a complex compound that may be an organic compound, maybe a petroleum product or something like that, if it is degraded using multiple steps, that is the compound A is converted to B, B is converted to further degraded or transformed to C and D and etc. And finally, it reaches a stage where the entire material or that compound is fully utilized and converted into water, CO2, and other things. So that actually is the term. That term is biomineralization, in which it is completely transformed into the mineral forms and further toxicity or that compound is not remaining in the environment. So biodegradation doesn't mean that it is the final target of biomineralization is achieved. So you can say that the entire traces of the compound and subsequent intermediates were removed only when it is mineralized. And simple biotransformations or biodegradations from a complex compound to a complex or even more toxic compound doesn't mean that the environmental threat is removed. So these are different forms of the remediation methods, and our focus will be based on biological measures. Okay, now we'll come to the next slide. And there are different categories of removal, or different groups or different types of removal. And the first one is a separation, which is actually based on physical separation methods, like soil washing. If the soil is contaminated, it can be washed with the water. That means including soil washing, stream piping, so steaming, then extraction based on vacuum or vacuum extraction, then solvent extraction, thermal desorption, particle separation. So these are different types of physical separation methods for the contaminant. Then there is another method that is known as containment. Containment means you are just anyway now in the age of corona, actually we are more familiar with containment. You are restricting that pollutant or the toxic compound within some confined limits, so that it will not spread to other areas. So there are different types of containments, and that can be stabilized within the containment using chemical stabilization methods, and you can use physical barriers or covers and solidification and vitrification methods, so that it will be confined to some area. Then hydraulic containment methods for PIM underground or water contamination management, hydraulic methods are there. Then removal methods based on landfill creation is there. So all this, you can collect all the hazardous materials and fill it in a landfill and contain in that area. So that type of measures are available for handling the waste materials. Then there is a destruction-based methods, which may include physical and chemical destruction. That is, incineration is an example of physical destruction. Then chemical destruction is based on dechlorination is there, glorification is there. Then another one, biological methods that includes natural attenuation and bioremediation based methods. All these are destruction of the compound, or we will completely remove, or finally the final target of mineralization is achieved during the destruction stage, especially through bioremediation. And all other methods, simple separation or containment, doesn't remove that, it is completely removed from the environment, and that threat is completely removed. But in the case of destruction, especially the biological methods of attenuations and or natural or removal of the toxicity and the final mineralization or bioremediation, which will subsequently lead to the biomineralization, is the final target in the removal of such a toxic compounds. Is it clear? Online, hello?
Yes, sir.
Okay.
Okay.
Okay, fine. Now, in the process of bioremediation, we have the microbial enzymes, which is doing the process. The microbes actually releases enzymes, extracellular enzymes, to break down that contaminant. Usually, the complex compounds were not as such absorbed, or it is taken, or it is imported into the across the cell membrane into the cell. And that means the microbial cell may release some enzymes. It may release some enzymes, extracellular enzymes, into the environment, which act on the compound, and further that after an initial stage of degradation, some of the organisms may absorb it, and along with the other, the contaminants will be uptaken by the cell membrane. Then oxygen and nutrients or other nutrients will also be taken, and it is treated as a substrate. And finally, waste and carbon dioxide is a mineralized, that is as well as in the mineralized form. This is how the degradation or the bioremediation works. The contaminant of organic substance, the contaminant of organic substances is ingest, or it is taken in and digested as a food along with the other energy sources by the cell. The goal of this bioremediation is to degrade organic substances that are hazardous to the living organisms and degrade the organic contaminants into inert products, and maybe in the mineralized form. So only harmless biological waste are all that remain of the contaminant. And at the final stage, only harmless remainings or biological waste. If it is completely achieved, mineralization is completely achieved, then we can say that it's waste because it is fully mineralized and CO2 and water is released. But in some cases, this biomineralization, in all the cases of bioremediation, this biomineralization may not be achieved. And one microorganism or a group of microorganisms, we may degrade a toxic compound into a less toxic compound, and still that biological waste may be harmless or non-toxic or less toxic. Biological waste will be expelled at this stage instead of the CO2 and water. So only in the case of biomineralization, the final compound released will be CO2 and water, that is the mineralized form. So, and what are the different factors that is affecting the bioremediation? The primary factor is bioavailability, availability of the compound for microbial action. Then second one, that is a physical and chemical factors that is involved in bioremediation. Then concentration of the pollutant, at how much concentration this pollutant is available in the environment for microbial action. Then and the final one is a recalcitrance of xenobiotic nature of the compound. And maybe you might have learned what is recalcitrance. Recalcitrance is the nature of the compound to resist biodegradation. And the compounds which is actually foreign to the biological environment, foreign to life. That is a xeno means foreign. Foreign to life, that is biotic. Xenobiotic is a compound which is actually a artificial or also a synthetic chemical or a compound which is normally not existing in the environment. It's not a natural compound. That is a xenobiotic compound. Maybe new compounds where complex compounds or maybe pesticides and other things which is actually artificially synthesized, that comes under the category of xenobiotic compounds. So the compounds actually foreign to life, and they shows the nature of a recalcitrance, or a recalcitrance means the tendency to resist biodegradation, that is shown by such compounds. So the degree of this recalcitrance or the nature of the xenobiotic compounds is a significant factor in deciding its biodegradability. The complexity or complex ring structures and other side grooves which is present, and the overall size of the compound, etc., are major factors that is designing the biodegradability of such a compounds. So when we talk about the bioavailability of the contaminants, the compounds may be fully trapped in the soil, or some slow diffusing materials will be there, and it's not fully trapped. Maybe it will be released in the soil. So in the soil itself, non-available materials are there, and then slowly available materials are there, rapidly available contaminants are there. This type of materials were present in the soil environment. Just a second. Okay. In the regarding the availability of these compounds in the soil, we have trapped, slow available, or rapidly available compounds are there, and these type of contaminants or the materials which is present in the soil may be available in the gaseous phase, that is concentration in the gas phase, then the contaminant in the gas phase, then contaminant in the water phase. In the soil itself, there will be water which will be carrying or taking out in the gas and water phase. In both the phases, these contaminants will be taken out, and subsequently through the biological membranes to the cell interior, this compound will be transported. So the final availability, anyway, we discussed the transfer of these materials when we learned the industrial microbiology, for example, in the gaseous exchange, the mass transfer. It's in a similar manner. The overall concentration which is present in the soil, the compound concentration which is present in the soil, will affect the concentration available in the gas phase, concentration available in the soil water phase, and finally, all these concentration will influence the available concentration or the transport of these compounds through the cell membrane to the cell interior. So there's a concentration from the high concentration. There will be the transport of these contaminants into the cell here, and these concentrations depend on the solubility of this compound at gas and water phase, etc. Decide how much concentration of this contaminant is available in the cell interior for the biological action or biodegradation. So the bioavailability of contaminants in the soil is affecting I would say affecting the degradation at all these stages. So you can say that the availability of the compounds in the soil at different stages, then its diffusion in the gas and water environments that is affected, that's affecting the final concentration or available concentration of the contaminant for degradation. Then the ability of that compound to pass through the biological membrane is another factor which finally decides the available concentration for degradation. So bioavailability of the contaminants is actually affected at various stages. Now, biostabilization is another term in biodegradation. And biostabilization is the biodegradation of accessible pollutant fractions in a soil or sediment matrix, leaving a bound residue that is much more biologically unavailable or immobile. So, as I mentioned, biodegradation doesn't ensure biomineralization. It may be converted into another compound. The compound a complex compound, may be converted into another, maybe a slightly lesser complex, a compound with a slightly less complexity, that is B. And that may be more unavailable or more immobile. And still, or more stable compound. B is a more stable compound and not available in biological terms. For example, the bioavailability of the intermediate compound B is much less. So it is immobile and is not available for further degradation by the microbes. In that case, we can say that the compound is biostabilized and it is made unavailable or immobile during the conversion process. So further degradation to the mineralization is not possible at that stage. So there's such as the term biostabilization is the conversion of a compound during the biodegradation to a more stable and immobile compound which resists further degradation. That is biostabilization. Now, the microbes that is used for biodegradation, that involves aerobic compounds, aerobic degradation, that is Pseudomonas, alkaligens, and Sphingomonas, that the organisms like Rhodococcus, then Mycobacterium, etc. These are all different aerobic organisms. And these microbes have often been pesticides and hydrocarbons, both alkane and polyaromatic compounds. Then many of these bacteria use the contaminant as a sole source of carbon and energy. So it uses that these type of aerobic organisms will use the compounds like hydrocarbons, alkenes, and polyaromatic compounds, and pesticides, even pesticides, as a sole source of carbon or substrate. So it will degrade them completely. Then there are anaerobic organisms, and not as frequently used as aerobic bacteria, and it is used for bioremediation of a polychlorinated biphenyls, that is PCBs, in river sediments, and dechlorination of the solvent, that is a trichloroethane, TCE, etc., and chloroform also. So for the degradation of PCBs, TCEs, and chloroforms, we have systems, especially in the sedimentary systems in the river, etc. We have anaerobic systems. Now, ligninolytic fungi like phenolocete chrysosporium, one example, and that is a white rod fungi. It is having the ability to degrade an extremely diverse range of persistent or toxic environmental pollutant, and that is a common as a common substrate used in including that stores, sawdust, or corn combs, etc. These are the different types of supporting substrates, like common substrates, including stores, sawdust, etc., on which the ligninolytic fungi will grow. And these lignin-based substances can be, or substrates can be degraded by these ligninolytic or white rod fungi like phenolocete chrysosporium. Then methylotrophs, another example, another category, that is a aerobic bacteria that grow utilizing methane as the carbon energy. Then the initial enzyme in the pathway for aerobic degradation, that is methane monooxygenases, has a broad substrate range and is active against a wide range of compounds. So the initial enzyme in the degradation is methane monooxygenase. In methylotrophs, the initial enzyme in the degradation is the methane monooxygenase, and it is having high range or a broad range of substrates. And it is active against a wide range of compounds, including the chlorinated allylic, then trichloroethane and 1,2-dichloroethane, etc. So a range of chlorinated aliphatic compounds can be degraded by these methyloctaves, in which the initial enzyme is the methane monooxygenase. So these are the soluble diion monooxygenases. When we consider the biodegradation of such complex compounds, monooxygenases, where different types of monooxygenases and dioxygenases are there, which is the actual enzymes that is degrading this complex compound into simpler forms. If you are asked to name a few enzymes that is acting on the xenobiotic compounds, monooxygenases were the primary ones. So we have a soluble diion monooxygenases, SDIMOs, and a lot of items were actually listed there. Different types of monooxygenases that is nowadays available, and these are the group of the soluble diion monooxygenases is presented there. They are, in short, it is abbreviated as a SDIMOs. And when we are using these monooxygenases in bioremediation, it is actually growth-linked metabolism. Means the substrate being attacked benefits the growth of the organism. So whatever substrate that is used in the system, or the contaminant is actually used as a substrate, and that contaminant will act as a substrate for the or energy source for the growth of the organism. That the growth benefits are derived from the subsequent metabolism of the reaction products. That means initial degradation of compound A to B may not yield energy, but usually the conversion pathway of conversion of the complex or the xenobiotic compound from A to B, then B to C and C to D. And during the pathway, it produces various intermediates, and that final degradation in the pathway, that subsequent metabolism of the breakdown products will yield energy. And it doesn't mean that in a single step reaction, it is directly utilizing, just like sugar, it is using the or it is creating energy, generating energy from the substrate. So the substrate means the substrate and or the compound or the toxic compound is degraded into similar forms in a various or multi-stage reaction, and second metabolism or the state at the different stages of the metabolism, energy is generated. So the overall result is that monooxygenases and their subsequent degradation products will generate energy or provide energy for the growth of the organism. Typically, enable these SDIMOs catalyze the energy consuming further degradation of the compound. So the further degradation of the compound actually generates energy. Now, next point is about co-metabolism. Co-metabolism is a case in which there is no benefit of the cell no benefit to the cell performing the reaction. That is if organism A is degrading something or converting a complex compound A into B by the organism, say some species of bacillus, or it is degrading complex A into B, but directly the organism or the bacillus species that is doing that job may not generate or may not get any energy. So there is no benefit for the cell performing the reaction. May retard the growth by competitive inhibition. But for many recalcitrant compounds, this is a useful option to initiate degradation by other process. So the organism that work on the initial conversion may not get energy from that, but usually after some stage, in that case, another organism will take over in the succession. And for many reactant and recalcitrant pollutants, this is a useful option to initiate the degradation process. So even though the initial organism is not getting some energy, or it is not directly using it as a substrate, still it converts the compound, and many oxygenases are very powerful that's able to insert oxygen atoms into chemically stable compounds. That's how these monooxygenases work at the initial stages. Then hydroxylation or epoxidation that renders it less toxic. So there are various processes in the uh, in the conversion, like the oxygenases will add or insert oxygen atom to the stable compounds. Then hydroxylation or epoxidation renders is less toxic and or more amenable to further degradation, or the recalcitrance nature is reduced during the hydroxylation process. Then SDMOs have a broad range of substrates or substrate range, and can insert oxygen atoms into a wide range of organic compounds. It not works on some symbol or a single compound. It is having a wide effect on a wide range of organic compound. It can insert oxygen and hydroxylation, and the net result is that the recalcitrant nature of the compound is reduced, so that it will be more amenable to degradation in the further stages or the next stages. So in co-metabolism, actually, the second organisms or the next organisms that are coming, or means it is actually done as a byproduct, or the organism will work on for the growth of the organism. It may require another substrate for normal growth. Co-metabolism, the organisms work on these toxic compound as not as a direct substrate, but the organism for its growth, it requires another normal substrates, and the enzymes which produce or the different enzymes that is produced by the organism will do this degradation as a side job. So it is not the primary objective of the organism, or the organism is not getting any direct benefit from the metabolism of this toxic compound. But the conversion is actually it is done along with the normal growth of the organism. The organism's primary objective is not the degradation of the concerned compound. Now, there are different benefits for the bioremediations. It is considered as less costly compared to the chemical and physical treatments. Then toxic chemicals are removed. It is not separated usually. Then the containment-based methods, for example, removal or containment-based methods, the toxic chemical is actually, it is removed or it is collected and gathered at some point, but it is not removed from the environment. But in biomineralization-based methods, what happens is that the entire toxic material is finally mineralized, and it is completely removed from the environment. That is the advantage of such a bioremediation methods. Then potential to be a dominant technology in the future. Various even commercial technologies were developed nowadays, and you might have noticed yesterday's syndicate decision. University itself is actually proceeding with some project on electricity production of 9 crores that's sanctioned by our university syndicates yesterday. The base materials will be generated. It will be used for methane gas production, and subsequently it is converted to electricity, and it will be connected to the electricity grid. So that proposal is actually approved by the university yesterday. So there are various dominant technologies which is based on such bioconversions and bioremediation, and in which the different forms of or different types of compounds can be converted into energy or fertilizers on another various useful products. So there are various technologies available nowadays. Now it can be used. It can use indigenous population. So you don't require normally you don't require a genetically engineered superbug like the Pseudomonas putida, which is uh, for the treatment of the crude oils. Indigenous organisms, indigenous populations can be used, enriched and used for a bioremediation purpose instead of going for such a genetically engineered ones. Then it leaves no toxic residues. If the complete biomineralization is achieved, then it will not leave any toxic residues compared to the chemical conversion techniques. Then suitable for in situ and ex situ remediation methods. In situ means on the site treatment, For example, soil is contaminated, or there is an oil spill, some leakage of the petroleum products. In all these cases, in situ, if it is the soil is contaminated, in situ treatment can be conducted on site. And ex situ means you have to carry the waste and aggregate at some point, then treat it there. So that type of treatment is the ex situ remediations. In situ and ex situ remediations are both possible using these bioremediation techniques. So they are the benefits. But still there are constraints also. Constraints means or limitations. There are some environmental factors. So the climatic conditions and other various environmental factors will affect the microbial growth. So in all stages and at all environmental conditions, you can't predict the same output of the microbial remediations. Especially in India, or we have some particular climatic conditions and environmental conditions. But at freezing temperatures, where in some countries, most of the times there will be freezing temperatures, and you can't expect such microbial action there. So that depends on the environmental factors. And another one, containment types. Uh, Based on the nature of the pollutants, for example, soil pollution or water pollution, whatever. And there will be different types of containments which is required for the treatment. And the level of the toxic compound or the nature of the toxic compound is also another factor. So we require different types of or different levels of containments for treating the waste. Then the nature of the organism that is involved, whether it's a genetically engineered one or a natural one, and whether it is having some environmental impact, etc. Then the cost benefit ratios, whether your technology is actually is a cost effective one. So if it requires a much higher cost for establishing and treating the waste, then usually it will not be sustainable, and does not apply to all surfaces. For example, you are treating water or groundwater, or different types of sources need to be treated. So all technologies may not suit at all. For example, bore wells are there, or petroleum wells are there, or even underground water sources are there. All these things were very contaminated at different points. So uh, these bioremediation technologies, all the technologies were not universally applicable. You have to design specific strategies for addressing such specific issues. Then the soil type, if the soil is contaminated, then the type of the soil is also important while deciding such a remediation techniques. Then another one, usually this compared to the chemical and physical processes, this is a slow process, and the heavy metals are actually, it is not removed. It's actually, it may be aggregated, but heavy metals cannot be entirely removed, or just like bioremediation, it cannot be removed from the environment. It will exist, but you can remove from some site, and you can aggregate and remove. That's all. Then complex waste need a multi-step process. Pollutant audit is necessary. For example, regular monitoring of the environment for the presence of the pollutants and the level of the pollutant, whether it is exceeding the critical limit, and then you need to implement some strategies for remediation. So you need a pollutant audit always. Then it requires a strong scientific base, monitoring site and engineering, or just like even for a biogas plant, you require specific scientific treatment for that one. If you dump everything in the biogas plant, within no time it will be spoiled. So you should have a scientific base while maintaining that one. So in larger treatment sites or treatment systems, then you need scientific understanding, then monitoring, proper monitoring, and site engineering for long-term maintenance of that system. Then not all quantities of contaminants can be addressed or treated using these type of methods. So there are positives and negatives for biological or bioremediation